Hello to everyone. In this presentation, we are going to study the remaining aspects of oxidation of fatty acids. In last presentation, you studied in detail the reactions of beta oxidation of fatty acid, its regulation and energetics. Now we will discuss disorders of beta oxidation of fatty acid. The first disorder is carnitine deficiency. Carnitine deficiency is commonly seen in premature infants due to decreased synthesis of carnitine or leakage of carnitine from kidney. This deficiency is also seen in patients of renal failure undergoing hemodialysis. You know very well, carnitine is absolutely essential for the transport of acyl coenzyme A from cytosol to mitochondria. Because of carnitine deficiency, there is no transport of acyl coenzyme A from cytosol to mitochondria, so there is no beta oxidation or decreased beta oxidation. And because of decreased beta oxidation, there leads to decreased utilization of free fatty acids, and that leads to increased plasma free fatty acid level. These plasma free fatty acids may undergo deposition in liver that leads to development of fatty liver. Because of again decreased utilization of fatty acid for energy purpose by muscle, that leads to weakness of muscle and myopathy. Again, there leads to excessive utilization of glucose for energy purpose, and because of that leads to hypoglycemia. Second disorder is carnitine transport defect. It may be due to deficiency of carnitine acyltransferase 1 enzyme or carnitine acyltransferase 2 enzyme or translocase defect. The most predominant defect is due to carnitine acyltransferase 2 enzyme. Because of the defect of any one of the component, acyl carnitine can't enter mitochondrial matrix, and if there is no entry of acyl carnitine in mitochondrial matrix, there is no or decreased beta oxidation. So there is no energy supply to muscle, so muscle becomes weak during prolonged activity, and muscle cramps are also commonly seen. These attacks are precipitated by starvation and severe muscle exercise. Treatment is avoid long chain fatty acid containing lipids and substitute these long chain fatty acid with short chain and medium chain fatty acids. Why you should substitute with short chain and medium chain fatty acid? Because for the entry of short chain and medium chain fatty acid into mitochondria, there is no requirement of carnitine transport system. So short chain and medium chain fatty acids can enter mitochondria and can undergo beta oxidation and can supply energy. Third disorder is medium chain fatty acyl coenzyme A dehydrogenase deficiency. It is the most common inborn error of fatty acid oxidation, found in 1 in 10,000 births. In this disorder, there is decreased ability of the body to oxidize medium chain fatty acids having 8 to 14 carbon atoms. So because of decreased utilization of these fatty acids accumulate in tissues. Symptoms which are seen in this disorder are vomiting, lethargy, hypoglycemia and coma. As medium chain fatty acids cannot undergo beta oxidation, these fatty acids divert to another oxidation which is known as omega oxidation. In omega oxidation, it produces small to medium chain dicarboxylic acid excreted in urine as glycine or carnitine conjugate. What is the treatment for this disorder? High carbohydrate diet and carnitine supplementation. High carbohydrate diet ensures uninterrupted energy supply, whereas carnitine supplementation will make sure that long chain fatty acids can enter into mitochondria and can undergo beta oxidation to supply energy. Many cases of sudden infant death syndrome are commonly associated with deficiency of this enzyme, that is medium chain fatty acyl coenzyme A dehydrogenase. Because of the deficiency of this enzyme, infant cannot utilize medium chain fatty acids which are present in milk. So they cannot derive energy from medium chain fatty acid, and because of that infant may die suddenly, and this disorder is known as sudden infant death syndrome. Next disorder is Jamaican vomiting syndrome. This syndrome is caused by eating unripe akki fruits which contain toxic amino acid hypoglycine. This hypoglycin A inhibits enzyme acyl coenzyme A dehydrogenase, which is the important enzyme of beta oxidation. Because of inhibition of this enzyme, beta oxidation is blocked. That leads to serious complications. Symptoms which are seen in Jamaican vomiting syndrome are severe hypoglycemia, vomiting, convulsions, and coma. After studying the disorders of beta oxidation of fatty acid, now we can see oxidation of odd chain fatty acids. Odd chain fatty acids are the fatty acids having total carbon 1, 3, 5, 7, 9 like that. So these odd chain fatty acids are oxidized in the same way as even chain fatty acids. What it means? Odd chain fatty acids also undergo beta oxidation, and in beta oxidation odd chain fatty acid produce acetyl coenzyme A and propionyl coenzyme A. If a 17 carbon fatty acid is oxidized, it undergoes 7 cycles like palmitic acid, which is 16 carbon even chain fatty acids. But here in the 17 carbon fatty acid oxidation, at the end 3 carbon unit as propionyl CoA is left instead of acetyl coenzyme A. So in oxidation of odd chain fatty acids, the end product which is formed is propionyl coenzyme A. Propionyl coenzyme A is having different fate. It undergoes carboxylation reaction by the action of enzyme propionyl coenzyme A carboxylase. In this reaction, ATP is utilized and biotin is required as a coenzyme. The product formed in this reaction is D-methylmalonyl coenzyme A. This D-methylmalonyl coenzyme A is converted into its L isomer, that is L-methylmalonyl coenzyme A, by the action of racemase enzyme. L-methylmalonyl coenzyme A is then rearranged into succinyl coenzyme A. For this reaction, vitamin B12 and mutase enzymes are required. Succinyl coenzyme A then enters TCA cycle and go for either oxidation cycle or for gluconeogenesis. Propionyl coenzyme A can form glucose after it is converted into succinyl coenzyme A and then into oxaloacetate in the gluconeogenesis pathway. This occurs in condition of hypoglycemia. So oxidation of odd chain fatty acid produces propionyl coenzyme A, and it is the only fatty acid which can form glucose through gluconeogenesis. Metabolic disorder of odd chain fatty acid oxidation is methylmalonic aciduria. This disorder may be due to deficiency of vitamin B12 or due to mutation in methylmalonyl coenzyme A mutase enzyme. Both vitamin B12 and mutase enzymes are required for the conversion of L-methylmalonyl coenzyme A to succinyl coenzyme A. So, if there is a defect in vitamin B12, the patients can respond to injections of adenosine cobalamin. But if there is a defect in mutase enzyme, these patients will not respond to injections of adenosine cobalamin. In this defect, methylmalonic acid accumulates in tissues, and that leads to metabolic acidosis, damage to central nervous system, and growth retardation. Treatment involves, you should avoid odd chain fatty acid and branch chain amino acid in the diet because both are responsible for the formation of propionyl coenzyme A, and later on its conversion to methylmalonic acid in the absence of vitamin B12 and methylmalonyl coenzyme A mutase enzyme. Unsaturated fatty acids are activated, transported across the inner mitochondrial membrane and undergo beta oxidation similar to that of saturated fatty acids. But the double bonds of naturally occurring unsaturated fatty acids are in the cis configuration, whereas those produced during beta oxidation are in the trans configuration. So, an additional enzyme called aryl coenzyme A isomerase is required to convert the cis double bond to trans double bond. When cis double bond is converted to trans double bond, it is recognized by aryl coenzyme A hydratase. Now the rest of the chain can be oxidized as described in beta oxidation. You can see here oxidation of monounsaturated fatty acid that is oleic acid, having 18 carbon in its structure and one double bond between 9th and 10th carbon atom. Oleic acid undergoes beta oxidation just like saturated fatty acid, and in 3 cycles of beta oxidation it releases 3 molecules of acetyl coenzyme A, and the product which is formed is cis delta 3 do-decaenoyl coenzyme A. This product is having one double bond between 3rd and 4th carbon atom. This double bond is in cis configuration, and because of that this product cannot undergo beta oxidation further. To enter in beta oxidation again, there is a requirement of enzyme called enoyl coenzyme A isomerase. This enzyme converts cis double bond into trans double bond, and it also shifts double bond between 3rd and 4th carbon to 2nd and 3rd carbon. So the product which is formed by the action of enoyl coenzyme A isomerase is trans delta 2 do-decaenoyl coenzyme A. This product now undergoes 5 cycles of beta oxidation and produces 6 molecules of acetyl coenzyme A. So it shows that monounsaturated fatty acid, oleic acid, undergoes beta oxidation just like saturated fatty acid with little modification and requirement of additional enzyme called enoyl coenzyme A isomerase. When polyunsaturated fatty acids having more than one double bond undergo beta oxidation, there is requirement of one more additional enzyme apart from enoyl coenzyme A isomerase. This additional enzyme is called 2,4-dienol reductase. You can see here oxidation of polyunsaturated fatty acid, that is linoleic acid, which is having 18 carbon in its structure and 2 double bonds, one between 9th and 10th carbon atom, and second double bond is present between 12th and 13th carbon atom. This linoleic acid, like oleic acid, undergoes beta oxidation for 3 cycles and 3 molecules of acetyl coenzymes A are released. After 3 cycles, the product which is formed is having 2 double bonds. One double bond is present between 3rd and 4th carbon atom, and second double bond is present between 6th and 7th carbon atom. Both the double bonds are in cis configuration. So this cis double bond has to get converted into trans configuration before entry into beta oxidation, and this is done by the action of enzyme enoyl coenzyme A isomerase. This enzyme shifts the double bond between 3rd and 4th carbon atom to 2nd and 3rd carbon atom, and the cis double bond is getting converted into trans configuration. So this product now enters beta oxidation and releases acetyl coenzyme A. After releasing acetyl coenzyme A, the product whichever is formed again re-enters in first step of beta oxidation of another cycle, and in that step the product formed is again having 2 double bond. One double bond is between 2nd and 3rd carbon atom, which is a trans double bond, and second double bond is present between 4th and 5th carbon atom, which is a cis double bond. Now 2,4-dienol coenzyme A reductase enzyme acts on this product and convert these double bonds into trans configuration and reduces one double bond. So the product is formed, and that product is having only one double bond, and that double bond is present between 3rd and 4th carbon atom, and this double bond is trans configuration. Now this double bond which is present between 3rd and 4th carbon atom is shifted to 2nd and 3rd carbon atom by the action of enzyme enoyl coenzyme A isomerase. After shifting the double bond to 2nd and 3rd carbon, this double bond is in trans configuration, so it can enter in beta oxidation smoothly, and 4 cycles of beta oxidation goes on and 5 molecules of acetyl coenzyme A are released. Now we can see minor fatty acid oxidation, which include alpha oxidation, omega oxidation, and peroxisomal beta oxidation. First we discuss alpha oxidation. What is alpha oxidation? It is a process by which fatty acids are oxidized by removing carbon atom one at a time from the carboxyl end in the form of carbon dioxide. So alpha oxidation process is mainly utilized for the fatty acids that have a methyl group at beta carbon. Because of the presence of methyl group at beta carbon, these fatty acids cannot undergo beta oxidation. And what is the example of fatty acid having methyl group at beta carbon? Phytanic acid. Phytanic acid is major dietary methylated fatty acid. It is also present in chlorophyll and animal fat. Its structure is shown here. It is having methyl group at 3, 7, 11 and 15 carbon atom. So this phytanic acid, because of the presence of methyl group at the third carbon, that is beta carbon, it cannot undergo beta oxidation and it has to divert to another pathway known as alpha oxidation. Alpha oxidation of phytanic acid is very important process in brain. If there is defect in alpha oxidation of phytanic acid, it leads to accumulation of phytanic acid in brain and subsequent development of Refson's disease. So to prevent the occurrence of Refson's disease, alpha oxidation of phytanic acid must be normal. This process occurs in endoplasmic reticulum mainly. It doesn't require coenzyme A and it doesn't generate energy also. Some fatty acids undergo alpha oxidation in peroxisomes also. What are the steps of alpha oxidation? Number 1, activation of phytanic acid by the action of phytanyl coenzyme A synthetase and utilization of energy in the form of ATP to form phytanyl coenzyme A. This activated phytanyl coenzyme A later on undergoes hydroxylation by the action of phytanyl coenzyme A hydroxylates, and the hydroxylated product later on undergoes breakdown to form formyl coenzyme A, which later on liberates carbon dioxide. Later on, pristanol undergoes oxidation to form pristanic acid, and this pristanic acid undergoes beta oxidation. So these are the steps of alpha oxidation. You can see these steps here in detail. We can start here that phytanyl coenzyme A is activated form of phytanic acid. This phytanyl coenzyme A undergoes hydroxylation by hydroxylase enzyme. Vitamin C and ferrous are required. Alpha ketoglutarate are utilized and alpha hydroxyphytanyl coenzyme A is formed. You can see here hydroxyl group is there at the alpha carbon position. Now this alpha hydroxyphytanyl coenzyme A undergoes breakdown by the action of lyase enzyme and formyl coenzyme A is formed. This formyl coenzyme later undergo breakdown to form formic acid, which liberates carbon dioxide. Now in this reaction, hydroxyl group present at the alpha carbon position is converted to aldehyde group, that is CHO group, and the product form is called as pristanol. This pristanol undergo further reaction by the action of enzyme aldehyde dehydrogenase to form pristanic acid. Now CHO group, that is aldehyde group, is converted to carboxylic group. This pristanic acid undergo beta oxidation and forms propanil coenzyme A and acetyl coenzyme A molecule. Metabolic disorder of alpha oxidation of phytanic acid is Refson's disease. Refson's disease is developed due to lack of phytanyl coenzyme A hydroxylase enzyme. This enzyme is also known as phytanic acid oxidase. Due to defect in this enzyme, alpha oxidation doesn't occur and phytanic acid accumulates in brain and other tissues. Phytanic acid is considered as toxic to ciliary ganglion cells and induces calcium-driven apoptosis in Purkinje cells. Recent studies have found that phytanic acid has rotenone-like action, so it inhibits complex 1 of electron transport chain and produces reactive oxygen species, which are reactive oxygen species are detrimental to the tissues, and this is the reason that neuronal cells and retina rich in mitochondria are prime tissues affected in Refson's disease. The patients present with severe neurological symptoms like polyneuropathy, retinitis pigmentosa, nerve deafness and cerebellar ataxia. Regression of symptoms is observed with restricted dietary intake of phytanic acid. And what is the source of phytanic acid? Milk. Milk is a good source of phytanic acid and it should be avoided. After alpha oxidation, now we'll see omega oxidation. Omega oxidation is a minor pathway and becomes active only when beta oxidation is defective. Substrate which can undergo omega oxidations are medium chain fatty acid and long chain fatty acids. Enzymes of this pathway are located in endoplasmic reticulum of liver and kidney. So this pathway operates in endoplasmic reticulum with the help of hydroxylase enzymes having NADPH and cytochrome P450. Omega group, that is methyl group present at the omega position, is converted to CH2OH group and subsequently oxidized with the help of NAD+ to a carboxyl group to produce dicarboxylic acid. Dicarboxylic acids further undergo beta oxidation to produce shorter chain dicarboxylic acids like succinic acid and adipic acid. Reactions of omega oxidation of lauric acids are shown here. Terminal omega methyl group of lauric acid undergoes hydroxylation by the action of enzyme oxygenase and form a product which is having alcohol group at the omega carbon. This alcohol group undergoes further reaction by the action of enzyme alcohol dehydrogenase and converted into aldehyde group. Aldehyde group later on undergo oxidation by aldehyde dehydrogenase enzyme to form carboxylic group. Now the product is having two carboxylic group at either end. This product undergoes many cycles of beta oxidation and form succinic acid and adipic acid. Now we can see the last fatty acid oxidation, that is peroxisomal oxidation. Very long chain fatty acids means the fatty acid having carbon atoms more than 22 are oftenly oxidized in peroxisome. That's why it is called peroxisomal oxidation. Oxidation in peroxisomes is similar to beta oxidation with significant difference in first reaction only. Acyl coenzyme A dehydrogenase enzyme produces H2O2 rather than FADH2. In beta oxidation first step FADH2 is produced, whereas in peroxisomal oxidation first step H2O2 is produced. This is the significant difference. The importance of peroxisomal oxidation is that very long chain fatty acids are degraded to octanoyl coenzyme A, which is having 8 carbon in its structure, and this octanoyl coenzyme A later on passed to mitochondria for beta oxidation. So long chain fatty acids are shortened in peroxisomes and later on passed on to mitochondria for beta oxidation for energy purpose. You can see here the difference between mitochondrial oxidation and peroxisomal oxidation. In mitochondrial beta oxidation, in the first step, FADH2 is generated, which later on passes to electron transport chain and energy is generated in the form of ATP. Whereas in peroxisomal oxidation, in the first step, FADH2 is generated, which passes electron to oxygen and forms hydrogen peroxide. This hydrogen peroxide later on undergoes degradation by the action of catalase enzyme to form water and molecular oxygen. So in peroxisomal oxidation, ATP is not generated, whereas energy which is released is in the form of heat. Metabolic disorder associated with peroxisomal oxidation is Zellweger syndrome. This syndrome is an inborn error of peroxisomal fatty acid oxidation. It is developed due to absence of functional peroxisomes in cell. It is developed due to defective genes encoding protein called peroxins, which are required for peroxisome formation. So very long chain fatty acids are not oxidized in peroxisome and they are accumulated in liver, kidney and brain. That leads to severe neurological deficit in patients, and this disorder is very fatal in first year of life. At the last, we will see one important justification question associated with fatty acid oxidation, that is, fat burns on the flame of carbohydrate. And what is the reason? Fat is broken down to fatty acid and glycerol that are oxidized via beta oxidation and glycolysis respectively to produce acetyl coenzyme A. This acetyl coenzyme A later on undergoes TCA cycle for its oxidation. Oxaloacetate formed from pyruvate by carboxylation supports and maintains this TCA cycle as a catalyst, and glucose is the primary source of oxaloacetate. So through oxaloacetate, carbohydrate fuels TCA cycle to continue as a flame in which acetyl coenzyme A, which is the end product of fat oxidation, is finally oxidized, that is burnt out. So it justifies that fat burns on the flame of carbohydrate.